Our last speaker in this session is one of our very own from the University of Saskatchewan, Professor Kirstin Bett. She's a professor of plant breeding and genetics in the Department of Plant Sciences, and is also a faculty member in the Crop Development Center. She leads the common bean breeding program here, and is also one of the leads in a very productive lentil breeding and genomics program. She's released several common bean varieties that are used extensively in agriculture in Western Canada. And you know, Kirstin spans from traditional breeding to molecular genetics and breeding and genomics, and in that area she's been the lead on the recent sequencing of the lentil genome that's opening up a lot more molecular genetic and genomic possibilities with lentils. And so today I welcome her to give her talk, Roles for Digital Phenotyping and Public Breeding Programs. Kirstin.
All right. So being last has its advantages because I can think about more of what I wanted to say, but at the same time, a lot of what I'm going to say you've heard already, but I'm going to try and put a bit of a different spin on it. I'm also going to warn you, I teach, so some of this may look like a lecture, sorry. Okay, this is just to get you started. You know, with students, you always have to put up a cartoon. So I'm sure, especially all the computer geeks in the room have all seen this one before. And I truly do live this all the time, and especially when the whole PERC project came up, it was like, oh, we can solve all your problems. And a year later, they're like, yeah, especially with lentil, yeah, maybe not. So we'll see. Just for those of you who aren't familiar with the plant breeding at University of Saskatchewan, it is one of the largest public crop breeding programs in the world. We have everything from cereals through to pulses and a few other things in between, and we've raised 400 commercial varieties since 1971, billions of dollars in profit to the producers and the ag economy in general. And just an example for, we like to trot out lentil, is the return on investment is estimated to be about $49 for farmers for every dollar they've invested. So, and it's a group of 12 breeders and 2 pathologists at this point. And I just want, and that's not just the Crop Development Center, there's a few of us not in the Crop Development Center who also do breeding. And all I want to point out here is this is a big operation. We are running it's a professional plant breeding programs, and as the previous 2 speakers have alluded to, these run pretty much like a machine, and if you want to interrupt the machine with new technology, you better have a good reason to be doing that, because first of all, the technicians will get on your case, and the last thing you want is angry technicians, and the breeders are like, don't get in my way with your crazy technology. You have to prove to me why this technology is going to make my life easier, my crew happier, and we're going to make bigger gains at the end of the day. So just as an example, one of the things that Van brought up was the idea of your sites and understanding your sites. So this is just a map of Saskatchewan showing the sites that we have pulse. This is just the pulse breeding program has locations. And for those of you not from Saskatchewan, some of those are a 6-hour drive. Okay, so you're gonna drive 6 hours and discover that was not the day to go take notes. Okay, and I don't mean because it was raining, I mean because the crop's not at the right stage or something's happened and your eyes on the ground, or maybe the local farmer who may or may not give you reliable information. So if you could get information, even if it were satellite, if we could get it quickly enough, we might be able to decide, are we taking that 6-hour drive or are we gonna wait? So just to lead into where all this fits, just a reminder what plant breeding is. We like to think of it as applied plant genetics. It is an art and a science. We always teach that, and until you've spent a day in the field with a breeder, you don't understand that art part. It's true. Every breeder will do something different, And Van Ripley just alluded to that. If you send different breeders in to take notes, they're all going to give you different answers, which drives the computer guys in this room absolutely wild, I'm sure. But it's just a factor of how we actually run the process. And breeding is a process. It's a well-defined process. Usually in breeding programs, we have this system, and we don't like to mess with the system. It's also very, very multidisciplinary, so we're getting input from all sorts of people. Usually there's a pathologist involved. Increasingly, we have entomology issues out here on the prairies. We have quality people, not that the people are quality, but that, well, they are, but they're doing crop quality for us, especially in the cereal programs. And so we get input, and we have to handle all this input. And Brian Rossnagel, for those of you who are familiar with Brian, always said, it's simple, you just keep the big bags from the green plots. And the combine is your ultimate phenotyper. Because really, does it matter how you got the high yield? You know, some days it doesn't within the breeding program. At the start of the breeding program, however, and this is where it gets more complicated, is it will dictate perhaps how you could get more yield later on if you understood how you were getting the yield. So that's why we're willing to entertain some of the plant physiologists in their crazy ideas. Okay, just another thing about plant breeding, it is a juggling game. So how many genes do you think are involved in yield? I would argue every gene in the plant has something to do with your yield, right? And so what are you actually selecting for? How many traits can you actually select for? How many traits can you handle at once in a program, right? And the other way of looking at plant breeding is that it's a lottery, because it's just a numbers game. The more tickets you have, the old ad, the more you play, the more you can win, but the bigger your chances of winning. Well, it's the same in breeding. The more tickets you have, which just means you need investment. Okay, you have to buy tickets, you need investment up front, you have to spend a ton of money to get to the end of the cycle and discover whether or not you actually manage to produce a variety that's better than what's already out there. And I would argue there's quite a bit of luck involved in this too, because it's truly a numbers game. And if you can't, for investment reasons or money reasons, can't put out a big enough population to actually find the winner, you haven't bought enough lottery tickets, you can't win. And just to illustrate this, because sometimes people don't actually understand how crazy this is. If you've only got one gene segregating, the whole perfect population size is like if you flip a coin twice, you're going to get a head and a tail, right? If you only have one gene segregating, you need, in a fantasy world, 4 individuals to catch all your phenotypes and genotypes, right? If you have 20 genes segregating, we're now up at starting to get into silly numbers, right? And so it's your perfect population size is 4 to the n on however many genes, never mind traits, because each trait probably has multiple genes underlying it. So what are you trying to follow, and how many individuals can you actually look at? And the way we end up having to do that is when we go through, and I'm only going to talk about inbreeding crops here, when we go from the F1 to the F2 to the F3 and so on, as you're inbreeding, at the beginning, you have to have lots of lines. And we're talking, and we've seen the numbers already from the previous speakers, huge numbers, in order to actually find the variability that you're actually looking for. Which in the case of pulse crops becomes a bit of a problem, because unlike canola, which throws thousands of seeds per plant, we're talking, I'm happy in common beans if I get 50 seeds off of a single plant. So trying to develop the population sizes that you would ideally have means that you have to play the game a little more efficiently. So early on, we're dealing with these easy to score traits, high heritability traits, you've heard this already. We're looking, and this is where the problems start in, especially in the pulse crops with small seed supplies, is you only have enough seed to actually do a small plot at the beginning. So our F3s, we have single meter by meter plots. That's all we can put out there. because that's all the seed we have, right? And then we move up a generation, you might be able to get enough seed for a long plot. In beans, we often only get one location. Sometimes you can get 2 locations. So are you really assessing the variability as it really truly is, or is the environment playing too high a level? The other thing that's different between your early generations where you have huge numbers is that really what you're doing is throwing away the utter garbage. You're not actually able to pick winners at that point. So really and because this is the generation that really sets what you have going forward, this is why we need to have the big numbers. And we keep as much as possible because you never know what interesting phenotypes are going to pop up once you get towards fixation. So then it's later on at the bottom half of the cycle, we're looking at these more complex, lower heritability traits because we can go to multi-locations, have multiple replications. We're looking at fewer individuals at this point, and we can really look at them carefully. So in pulse crops, because they're also big, we tend to have 100, 150 crosses per year. For each of these, you're going to get not as many as you would like, but the most we can get. F2 plants coming off of each of these F1 plants from the crosses. And really, at the beginning in pulses, it's, as Pierre Huckel calls it, a beauty contest. We are going in and one of the nice things about the pulse is that color, seed size, and seed shape are very important, and they are fixed really easily. So we can do a lot of culling right at the beginning just by looking at things on the table like this. And we will put out thousands and thousands and thousands of seed samples and just throw them away, most of them. Not most of them, but a lot of them. And then we take them out to the field and have a look at them in these meter square microplots. Where you're at a single rep, we may have enough seed to go to a disease nursery if we're lucky. And as you can see, these are not lovely corn plants. So we're looking at, and they're not even soybean plants, which are, I hear, difficult to phenotype. We're now looking, this is not beans, this is lentils. And they're kind of ugly to phenotype because they don't grow on a big stalk with big leaves that you can see individually. It's just a big jungle in there. So that's what we're up against for phenotyping that. And then, so one of the things I was thinking about, where would what I would really love as a plant breeder, and it comes up in the winter, you're sitting there and you're looking at the data that you collected like this, and then you think, yeah, I kind of vaguely remember being in those plots, and I remember there was a problem, and is that why this one line isn't doing as well as I had thought, or what are these cryptic notes I've typed into the tablet and usually there's typos and you just think there's got to be a better way. And one of the things that so this is what we're making selections on in the winter is a bunch of numbers, a bunch of cryptic notes, and some seed that's in front of you, and you have to pick the winners out of this, right? So if we could have, obviously more information would be better, and even if all I had, and I've asked one of the guys on our crew who has the drone now, could you just take a picture of every plot, or every trial, so that in the winter I can pull it up on my screen and go, oh yeah, I remember walking through this field and I remember what was going on at that time. All right, you've seen this equation in multiple different fashions from the last 2 talks. Are you sick of it yet? But it is the most important equation for plant breeders because it's the response to selection. And the biggest, and I would, yes, your population size is important. Yes, the amount of genetic variability you have is important. Those are easy things to fix, right? But the heritability is the part that can give you probably the biggest bang for your buck once you have your population. And this is because it's detailing how much of that phenotypic variation you're actually seeing is due to genetics, because there's no point in selecting on phenotype if it has nothing to do with genetics, right? So we want to understand the underlying genetics of it. And so the heritability of a trait is going to determine your phenotypic method. When are you going to do it? Can you eyeball it and just give it a rating of 1 to 9 or 1 to 5 or 1 to 3 even? Or do you actually need a measurement like this many grams per plot or something like that? Can we get away with replication or not? If it's high heritability, obviously you don't need to replicate. So the problem is in early generations, if you only have one replication, how much do you trust that data? And is it just too bad that one particular individual ends up in the crappy end of the field? So this is one area that I'm really interested in exploring, is the idea of accounting for your environment. So this is just a couple of images that I got from Steve Shurtleff. This is actually wheat from Curtis Pozniak's program, but it's just to illustrate that on the left, you can see it's a digital surface map. So forget that there's any plots in there. It just shows you it's sort of like a topographical map, and you can see on the left that that there's a low spot. So in a year, if we continue to get rain like this, the plants that are down in the low spot are probably not going to do as well as the ones up on the high, and in a dry year, maybe they'll do better. So when you take your yield or you take your heights or whatever phenotype you're after, is that truly what genetically that plant is worth, or is it being masked by that horrible environment that it's managed to land in accidentally, right? So if we can actually account for the variability in the field somehow in making our decisions, perhaps we could actually get a better handle on the genotype rather than the phenotype when we make those selections. And you can see that in the NDVI on the right. You can see that there's variability within those plots later on. This is just before harvest. So what does that actually mean? And if we can actually relate it to the environment, maybe we'd be better off. All right, the other idea is that of indirect selection. So this is when you're selecting on another trait in order to get your trait of interest. And that could be, and mostly it's done because the secondary trait is much more heritable than your primary goal trait. And it relies on the fact that there's correlations with the trait you're interested in, hopefully high correlations, and the heritability being better. And this leads to two ideas. One is a lot of us are familiar with the use of marker-assisted selection, where markers' DNA has a heritability of 1. And so obviously high heritability, and as long as it's strongly correlated, preferably right in the gene you're selecting for, that's great. Or with genomic selection, it's correlated somehow with your trait of interest, and you can select for it. The other way of looking at it would be can we use digital images as your indirect selection? So we're picking something out of the image that somehow is correlated with your final trait that you're interested in, and that might make things a lot easier. Here's just a couple of examples that we've been running. I had a grad student who's killing himself trying to take data from herbicide trials, and what happens, it doesn't kill the plant, this herbicide, it actually just knocks it back, and then they regrow. And so then you're out there and you're trying to score, and you're not sure, well, is that because of how do I score this? And then as you're walking through this big nursery, and by the time you get to the back, you're scoring it totally differently than you were at the front, right? Same thing happens in disease. When you're at the front and you're fresh, you're doing really well, you get into the back and the mosquitoes are killing you, and you're just quickly trying to take data, and it's really, is it meaningful anymore? And so one way that we would really like to see some of the imaging is to extract the information. It's there, it's objective, not subjective, like some of the ratings tend to be. And one thing I'd like to point out is that we need to be sure to distinguish between physiology and all the fancy stuff, and not that what you're doing, what we were talking about earlier this morning doesn't hold up. That's really cool, and there's potentially some really interesting technology that will come of it. Obviously, there's going to be interesting technology come of it, but let's not confuse physiology and genetics with plant breeding, which is slightly different. But where they tend to come together, especially in a public breeding program, is the whole area of grad student projects, because training as a plant breeder does not make for a thesis. So we always have to throw projects at the students, and this is where the fun stuff happens. And I think we've got some fun projects going on with the students right now, especially with the digital imaging. And what I throw out to them is our goal is to make cheaper, easier, more effective methods for phenotyping for plant breeders. Okay, and until you can prove to us that it's cheaper and easier and most importantly more effective, we're not going to pick it up as breeders. So that's where the goal of the projects then is to actually answer those questions. The others would be doing projects on molecular marker discovery, and in order to identify molecular markers, or better yet, the candidate genes, you need to have accurate phenotyping. If you can't phenotype it properly, you're never going to find that gene, right? Or find a marker that's very useful. You need the best phenotyping possible. And the other area that a lot of students are getting into now, especially with a lot of them have better computational skills now than they used to, than we used to, for sure, is new methods of looking at data. And are we actually making our decisions based, on kind of fly by the seat of your pants decision making? I feel that some days it is. Versus you're actually incorporating all the information that you can into a model that will say, no, this truly is better than this other line. Even though on the surface it may not look quite like it, or they may look equal, if you have ways of incorporating the environmental data into your analysis, you may actually come up with the winner more reliably. And then the last area is this whole area of pre-breeding, the use of wild relatives, for instance, how not to shock the system when you introduce this novel variation. Because, yeah, you can cross with something unadapted, you are going to get a whole whack of variability, which in theory should raise your response to selection based on that equation, but your heritability is going to go out the window because you can't assess enough individuals to find the winners. And so my take-home message from the public, breeders, from my perspective anyway, is that breeding's a numbers game. We're selecting on phenotype, not genotype usually. And the more we can account from the environmental variability, the more effective this selection is going to be. And we think of it, mostly what plant breeders do at the end of the day when we're picking our winners is it's a mental index selection for many, many traits. And perhaps a picture may be worth 1,000 words, and that we would be able to incorporate that into our decision-making processes. So that's all.